Dan is both a philosopher and a biologist. He has a dual training. He did his PhD at the University of Exeter with Lenny Moss, who is a well-known philosopher of biology. And then he did his postdoc training, and so recently he worked in a very well-known theoretical institution, the Konrad Lorenz laboratory in Vienna. And I think now he's just in between the Konrad Lorenz, and he will start soon as a professor at George Mason University. Research subject of Dan for a long time has been mechanism. He's been working on mechanism in biology and from a critical point of view. So he wrote several well-known papers on the subject. And this is, so you can understand his title. This is exactly what he is going to talk today to us.
First of all, I want to thank Jean-Jacques and the organizers of this conference for inviting me. It's a very sort of unusual event, very interdisciplinary. I'm enjoying it a lot so far. So I'm a historian and philosopher of science, as Jean-Jacques has mentioned. And I'm going to be sort of trying to offer my 2 cents on what is interesting and exciting about the renewed interest in stochasticity and biology by looking at this problem of biological ontology. Now, this is what ontology is what philosophers refer to when they are talking about the nature of things, right? So a lot of my work has been focused on, you know, this very fundamental question in biology of what is the nature of life, what is the nature of living systems, okay? And since the 17th century, the very sort of predominant view, or the answer to that question of what living systems are, has been that they are machines, okay? Complex machines, and so this is where the notion of the Bête-Machine fits in. It's, of course, a notion that we can trace back to Descartes, but it's a notion that has been extremely influential and successful. And so really, what I want to suggest today is that the reason why we should pay more attention to randomness, the reason why stochasticity is philosophically interesting is because it challenges, perhaps mortally challenges or damages this notion that's been so important for the last 300-400 years in biology. So this is just a quote from a very well-known, distinguished evolutionary biologist, Dick Lewontin, not so long ago, 10 years ago, 13 years ago, just pronouncing that really biology, present-day biology, is the realization of the famous metaphor of the organism as a Bête-Machine elaborated by Descartes in part 5 of the Discourse. And so the idea, I'm sure you can already sort of understand, is that the difference between mechanical systems and biological systems is only quantitative, it's not qualitative. We can use the same sort of principles and methods and ideas that enable us to understand machines to understand biology. So really there's no need for an autonomous biology, there's no need for getting all worried and upset about the mystery of life, because ultimately we can reduce that mystery to mechanism, to complicated clockwork, okay? That's the idea. You see it everywhere today. Just to take another very famous, perhaps the most famous biologist at the moment, maybe not for the best reasons, but nevertheless, Richard Dawkins, in his Selfish Gene book, saying that we animals are the most complicated and perfectly designed pieces of machinery in the known universe. And this is not something that you only find in review articles or in in textbooks or in popular science books. You also find in technical articles. And in the context, I'm going to be focusing specifically in the context of cell and molecular biology, but you can find it also, of course, in evolutionary biology, developmental biology, but for the purposes in the interest of time, and just to keep my discussion focused, I'm going to be looking at specifically how this notion has influenced and really dominated the way we think about cells specifically, okay? So, I mean, the idea of thinking about life as mechanical is very old. You can quite easily trace it all the way back to the advent of modern science and the sort of the mechanization of the world picture that takes place during the scientific revolution, right? Which is a process which really gave rise to what we call now modern science, that required us to reconceptualize, to change the image that we had about nature, and think of it not in some sort of animistic way, but as a mechanical sort of piece of clockwork that can be understood, and that actually everything in nature is also mechanical. It's not just that nature's mechanical, but organisms being part of nature are also mechanical, and consequently we don't really need a wide range of sciences and disciplines, just that we actually can use mechanics as a foundation for basically everything: physics, medicine, biology, chemistry, and so on and so forth. So philosophically, it's really, we're talking about a philosophy of nature. Mechanicism is, as I've said, the idea that nature can be understood ontologically as a machine, nature and everything in it. It also comes with a range of other commitments. Reductionism, which I'm sure you're all familiar with, the idea that the way to understand the system is to break it down into its constituent parts, study those in isolation, and then build up a view of the whole by sort of conceptually reassembling the knowledge that you've generated from the parts. And determinism, right? The idea that everything you find, every event in nature is sort of predetermined by a previous cause, which really leaves, of course, no room for chance. There's no room for randomness or chance in the worldview that gave rise to modern science. That's something to bear in mind, very relevant, not just for biology. The view that we have of what constitutes good science in the 17th century hasn't been one that has led any room for chance, at least until the 20th century. Chance was just taken to be a sign of our ignorance. We didn't know the causes, we referred to them as random. But there was always the assumption that there was something, that the causes were there, just that we didn't know them. And importantly, I just want to draw attention to the fact that the commitment to reductionism is grounded in the machine ontology. It is because we think that systems are mechanical that we feel licensed to break them down into parts and not worry that we're going to lose any information about the whole by doing that. Okay, so it's a way of grounding that methodological approach. It gives us a way of dealing with nature if we think about it as a machine. And similarly with determinism, if the idea that nature is regular, that we can mathematize it, that we can predict it, is again grounded in the notion of a machine. If you eliminate that ontological assumption, it's very difficult to continue to sort of justify any sort of deterministic view. I mean, I'm not saying it's not possible, but at the very least, it just makes life easier, and it just gives rise to this sort of very coherent picture, right? And so that's important to also bear in mind. If you look at the history of biology, what you find really is that it's a history of the development of the machine conception of the organism from the 17th century onwards, right? So beginning with this sort of clockwork view, which was championed by Descartes, although of course Descartes' view is more complicated because he was inspired by water automata, pumped, sort of fountains pumped by water. So it's not necessarily just the clock that is important, but the idea of having parts that are coordinated, that work together in a coordinated way to produce a predetermined outcome is the notion here. And then by the 18th century, with the advent of the Industrial Revolution, you have a sort of a new way of thinking about nature and about organisms as a sort of a steam engine, right? Where you have a transformation of energy. Of course, this is the time when thermodynamics develops, and so actually thermodynamics develops very much also not just to understand energy conversions in physics, but also to understand the conversion of energy into work in physiology. Lavoisier, to take another Frenchman, I'm trying to use as many Frenchmen as I can in this talk, historically speaking, to show that this is a tradition that's very important in France as it isn't anywhere else. Lavoisier actually defines the animal as a sort of a steam engine with the capacity to regulate and respirate, to breathe, and where you have this sort of conversion of energy into work. And then when we arrive at the 20th century, you have this view of the organism as a computer. The sort of the machine of the 20th century, really the paradigmatic machine, especially of the second half of the 20th century, is that of the computer. And so again, that technological transformation gives biologists a new handle on life, because we no longer have to only rely on mechanical analogies or in sort of thermodynamic analogies. We can also start thinking about organisms as systems that process information, that execute algorithms, and we can think maybe of development as that kind of process, or even as evolution as that kind of process. So, and a good example is François Jacob, who actually coined with Monod and Ernst Mayr the concept of a genetic program in 1961. Because you got this very sort of interesting history of the machine conception of the organism. And if you look at the, of course, the idea is that this notion is not just relevant at the organismal level, it's also relevant at every level of biological organization. Another Frenchman here, Changeau, is saying that the analogy between an organism and a machine holds true to a remarkable extent at all levels at which it's investigated. Good example of this is if you look at any sort of work in synthetic biology, you have these analogies being made at different levels of organization. And so really it's not just some sort of some loose metaphor. We're here using the notion to ground our conceptualization of what life is. It's playing a very fundamental theoretical role, right? So it's not enough, you should not be convinced if someone says, well, this is it's just a metaphor, because that just shows a misunderstanding of the importance of metaphors in how we, in science and how we develop language. So, the fact that the machine metaphor is so broad and so diverse and has such a rich history means that it's extremely versatile and is very, scientists can use it in different contexts for different purposes. Sometimes the reference is to some sort of cogs and wheels. Other times the reference is more to some sort of computer system with software and hardware and information transfer, information control. And if you think about molecular biology and about the philosophy of molecular biology, here I'm going to refer to another Frenchman, Morneau, in his Chance and Necessity from 1970. He really tried to make the case that molecular biology represents the return of mechanicism, if it ever went away. The idea that all the commitments that we had in the 17th century are commitments that we see today in molecular biology, and that unless you are committed to these views, you can't really understand what molecular biology is all about. So just to take the three principles that I mentioned, you find very prominent molecular biologists could be, showing an explicit commitment to these views, be it Morneau or be it Crick in relation to the question of reductionism, saying that the aim of molecular biology is to explain everything in terms of molecules. Or in the case of the question of determinism, the idea that if you have the complete sequence of DNA of an organism and a large enough computer that you would be able to compute the organism. And this is obviously a reference to Laplace, another Frenchman, the Laplacian demon that would be able to predict the future from complete knowledge of the initial conditions and a reference to the laws of physics, right? So these ideas are all very much alive today. They haven't gone away, and this is, I think, quite an interesting situation to find. So as you might have, I don't know if you suspected this, but I think John Jack already mentioned, a lot of my work over the past decade has been to punch holes in that sort of conception, show that it's problematic in a number of different ways when used in a number of different contexts. And I'm not going to try and tell you, try to summarize, my views on this. I'm just going to focus specifically on the question of the cell. What's the problem with thinking about the cell as a machine? This is actually a reference. It's not, I've made this up. This is actually a book published last year by Cambridge University Press. Again, just to show you that I'm not developing sort of a straw man here that I can then flick and pull down. This is very much a conception that practicing molecular biologists subscribe to. So what's wrong with the machine conception of the cell, you may ask? There are a number of serious problems, some arguments that can be developed against this view are more sort of philosophical, looking at the notion of teleology, and I won't discuss those. Some relate to thermodynamics and to the fact that organisms are far from equilibrium systems that have to constantly intake energy and that are sort of dynamically stable in a way that machines are not, because machines tend to be at equilibrium or in near equilibrium conditions, which sort of really changes the way we think about their organization. I'm not going to talk about that either. I'm going to use the third sort of sets of claims which can be brought together under what I call the argument from scale, okay, which is what I'm going to talk about today, about why we shouldn't think about cells as machines, cells and their parts, of course, not just cells as wholes. And just the point that this is a kind of what I'm going to present to you is not new. It's actually really old. It was first, I found it first formulated in Darcy Thompson, a very prominent biomathematician, Scottish biomathematician from the beginning of the 20th century, who wrote this very, very famous work on growth and form, where he tried to apply physical principles to understand biology. Most people remember him for his ideas about transformation, because they relate to evolution. But the chapter that I find most interesting is chapter 2 of this book. It's called On Magnitude, and the whole chapter is about how physical forces exerts a fundamental influence on the kinds of conditions that are available to organisms, and that depending on the scale, right? Depending on whether you're a large organism or you're a small organism, if you're an elephant or if you're a mouse, you're basically subject to sort of different forces are important to you, and consequently, they're going to determine and shape your anatomy, your physiology, your the way you behave, okay? That's the idea. And I just want to have this quote here for you to see this is actually the last paragraph of that chapter that I mentioned, where he actually is talking about different worlds. It's not just that they are subject to different forces, they are basically living in different worlds, right? He says, the world where the bacillus lives, right, gravitation is forgotten, right? Gravity is not important if you happen to be a bacterium because you're too small. But other things are important, right? The liquid, the water, your medium is no longer really fluid, it's more viscous. You have all sorts of forces that are important. Most importantly, of course, Brownian motion, which has come up a couple of times already in previous talks today, is inevitable. Thermal agitation leads to the constant jittering of everything, all molecules, and that is basic. That conforms the environment that you exist in if you happen to be a molecule, a protein, or if you're a cell. Not just Brownian motion, of course. Darcy Thompson mentions electric charges, so the ionized medium, so you can think of van der Waals interactions and all sorts of other electrostatic forces that are important. But the important point, he says, the predominant factors are no longer those of our scale, right? We have come to the edge of a world, he says, of which we have no experience, and where all our preconceptions must be recast. We can't just project onto that world the way we think about our world, because the physical forces that matter to us are not going to be important to the entities that we want to explain, okay? And I think this is a brilliant observation, and unfortunately one that molecular biologists sort of forgot, at least for much of the second half of the 20th century. So my argument is basically building on that observation of Thomson. Differences of size and in the physical scale in which organisms live are of overwhelming importance in determining their structure and behavior. By virtue of the microscopic size, cells and their molecular components are subject to very different physical forces than macroscopic objects. Okay, I've said this already. And that really we can think of microscopic and macroscopic organisms as inhabiting different worlds, whereas the macroscopic world, our world, and the world of our machines, the ones that we build, is ruled by gravity and inertia. The microscopic world of the entities that we want to explain and we are studying as molecular biologists is dominated by Brownian motion and diffusion. It's a different world. Consequently, our intuition, which is based on our experience of the microscopic world that we live in, is bound to lead us astray, is bound to fail when judging the adaptive problems that cells have to overcome. We're going to not be very good at understanding what cells have to do or what molecules have to do to perform their function if we are thinking about them the way we think about our machines and the way we think about ourselves, right? So, what implication does this have? Well, the implication is that we should be extremely skeptical of analogies, right, that seek to explain the microscopic world by appealing to the properties of macroscopic ones. But this is precisely what the metaphorical appeals to machines in the explanation of cellular phenomena attempts to do. When you hear about, engineering approaches, when we think about circuits, and when you think about, engines and all these sort of mechanical analogies, what we're doing is exactly this, right? We're sort of committing this mistake. But why do we do it? Well, we do it because they're familiar. I mean, we need to use something to make sense of what we don't know. What do? We appeal to what we do know. What do we know? Machines. Why do we know machines? Because we created them. And so we understand how they work. And it's comforting to us to know that they work in a regular way. And so we are sort of wanting to look at the world in this mechanical way. It's almost like we're predisposed to it. And yet, this is very problematic and ultimately is an untenable situation, because if machines were the size of molecules, they would not be able to function the way that they do. Now, you may be asking, what about this idea of nanotechnology? And we can talk about this in the Q&A. I don't really have time to talk about this, but the idea is simply that the macroscopic machines that we use to make sense of the biological world are going to fail us when looking at microscopic phenomena, okay? That's the argument. Right, so let's have a look at what's going on in molecular biology. Well, Since the 1990s, actually, since the publication of this paper by Bruce Alberts, who was then the president of the National Academy of Sciences, where he published this sort of manifesto called The Cell as a Collection of Protein Machines, preparing the next generation of molecular biologists, there's been a flurry of interest and a sort of propagation of this notion of a molecular machine as it's been basically extended to refer to all ranges of subcellular assemblies, right? The language of machines is everywhere. And it's also, again, as I've mentioned, it's super diverse. The way we biologists appeal to machines is never exactly the same. Different sort of analogies are proposed to make sense of biological systems. Now, you may think that this is inevitable, and I don't know. I think that the use of metaphor is probably, an analogy is probably inevitable, but we need to be extremely careful about the ones that we choose to use, right? Because they come with all sorts of connotations that can lead us astray. So what's the rationale? I mean, why do we think that this is a good idea to think of a protein complex, for example, as a molecular machine? Well, because if you look at the history of molecular biology, what you find is that there's a sort of a confluence, a convergence of two separate schools. One which tends to be called the informational or structural school, and the other one called one called the instructional school, the other one called the informational school, right? I won't go into the detail here because I don't really have time, but the point is that though these two schools came together in a spectacular fashion with the elucidation of the double helical structure of DNA by Watson and Crick in 1953, which required both of these things. It required thinking about structure, right, and also required thinking about specificity and information, how information can be coded in that structure. But the interesting thing is that these two sort of interests or emphases in molecular biology continued after 1953, and they sort of focus on different things, right? So the structural school of people in that tradition think that structure is important because that's the way we learn about function. Structure gives us a means of understanding function, and that leads to what sometimes is called the central dogma of structural biology. This isn't the central dogma of molecular biology. It's a different one, simply referring to the fact that the sequence of amino acids determines the structure of the peptide chain, and that structure determines the function, right? And then the other school, informational school, emphasizes this idea of highly coordinated operation. That there is the operation of molecules is very, very specific, and that the environment can be sort of cast aside. It's sort of context, and for the most part, we can focus inwardly on how the system, how the parts of the system, work in a coordinated way without worrying about influences from the outside. So if you look, this is just a quote from the paper that I mentioned, the Alberts paper, and here Alberts is explaining why we're using the molecular machine concept, right? He says, asks, why should we use the notion of the molecular machine? Well, because, and here is the analogy with the human machines, these proteins have highly coordinated moving parts. Within each assembly of, there's lots of constraints, right? All possible intermolecular collisions are actually eliminated, and they're restricted to a small set of possibilities, right? And so I could, give you other quotations from people focusing on why the focus on structure enables us to think about machines, right? That it is because these proteins can be understood from the structural perspective that it is appropriate to think of them as machines. So what's the problem? Well, the problem is that we don't really have a proper analogy here because they're very, very small. Because they're very, very small, these assemblies cannot possibly operate in this orderly, reproducible manner that we expect from them, from my idea of macroscopic machines, right? Do you remember, in a machine, as I say here, the motions of the various parts are orchestrated, right? So, for example, when a gear rotates, the shaft to which it is connected rotates in synchrony, a spring is compressed, the latch is released, you've got this sort of nice mechanism, right, which enables us to understand the operation of the whole. And all these movements are purposeful. They are being designed to be placed where they need to be to do what they have to do, right? They're predictable. We can say in advance what's going to happen, and that process always happens in the same way. And so you've got this very nice sort of at least intelligible means of understanding complex behavior. The problem with projecting that to cells or to proteins or to molecular processes is that, as I've already mentioned, macromolecules are constantly subject to continuous Brownian motion, which means that the majority of these, for example, conformational changes that proteins undergo are actually not the result of some sort of predetermined design in the genetic program, but are rather random walks that have nothing to do with their function, okay? That's the problem, and I just want to focus on be more specific. and focus on the two things that I said the molecular biology concept reunited. So the notion of rigidity or structure and specificity. So let's just focus first on structure. Why is the molecular machine concept problematic? Because it overemphasizes the structural rigidity of proteins in a way that is not physically realistic. Again, a simple reason for why it is not realistic is because of the scale that we're dealing with. And any physicist will be able to understand this, right? By virtue of their size, I say, proteins cannot exhibit the sort of rigidity and specificity that characterizes the structure and operation of machines. Actually, they're not even solids, right? I mean, so interesting recent work suggests the way we should think about the state of matter that proteins exist in is something closer to dense liquids or melted solids rather than a sort of a solid cog in a machine, right? And of course, if they're liquid and obviously it's much harder to imagine them being rigid. And it's also important to realize that most proteins don't have a single function, a single sort of structure, a single conformation. Rather, they exist in a state of flux, right, where they are constantly stochastically sampling a range of possible conformations, and then moving to the one that is most energetically stable. So if it was the case that most proteins had an energy profile like this, then we would probably have a situation where you can determine the sort of conformation on the basis of the amino acid sequence. But actually, a lot of proteins have energy landscapes that look more like this, which means that proteins can sort of sample this landscape, or find themselves in different places of the landscape, and as a consequence have different conformations, which means that you can't predict the conformation on the basis of the amino acid sequence, right? So this is what's led to the idea that many proteins are intrinsically disordered, meaning that they don't have a clear ordered conformation. They rather exist sort of as loose peptide chains that then, adopt a conformation when they happen to find a binding partner. Okay, so all context is obviously everything in that context, right? Because you can't just assume, you can't understand what the shape is going to be just by looking at the amino acid sequence. Similarly, with functional specificity, we now know that proteins actually are not as specific in their function as we thought. Actually, many of them perform different functions depending on when the protein is expressed and where, and depending on the part of the peptide that is active. And so this has led to the idea of moonlighting. Moonlighting proteins that do different things when no one's looking, kind of thing, right? So, and this is problematic because many proteins are labeled by their function. And it turns out that enzymes can also perform structural functions and so on and so forth. So again, we've got a problem here with thinking about this linear sort of sequence structure function idea, and we have to sort of come to terms with the fact that protein interactions are non-specific, most of them at the very least, that the interactions between proteins are short and caused by stochastic events. Okay, and so these associations are much more transient than is often assumed. And really, the fact is that proteins exist in this sort of environment where the only way they can interact with one another is through these probabilistic collisions, right? This was actually nicely shown by Andrea Paldi in his talk. And again, what I want to say here is that all of this is just a consequence of them being very small. And so what you have then is these sorts of appeals to circuitry, which you tend to find in as posters, in biology faculty buildings across the world, are extremely problematic because they're giving us a view of the cell. This is actually the map of all the sort of metabolic pathways, which is very, very sort of misleading. Here all the nodes, this looks like a solid state circuit. All the nodes here represent proteins, and the wiring is actually information transfer. Obviously, this is comforting because it gives us the illusion of control. It makes us feel good because it makes us feel that we understand what's going on here. But it's a very misleading view of what's going on in the cell due to the simple reasons that I've given to you. Due to scale. Due to scale, you cannot expect reactions to occur in this way. First of all, because there's no solid physical wiring connecting the nodes. Secondly, because these nodes exist in a sort of Brownian storm, which prevents us from making these sorts of characterizations, right? So I'm not saying it is impossible for molecules to interact in this way, but that this only represents one of many possible ways in which each of these molecules can interact, due to the fact that, as I said before, they're much less specific than we thought. So, this becomes really problematic when you focus on, you narrow down on a little piece of this map, right? And then try to work out empirically, experimentally, how often you find sort of interactions across different pathways. This is what a group did and ended up with this, what they called a horror graph, which is very revealing. The black arrows show the sort of textbook representation of this signaling cascade, but the green and red arrows show all the other ways in which these proteins have been found empirically over the course of just 2 years of looking at the literature to be engaged in, right? So it just shows that these sort of representations are more likely to mislead us than to enlighten us. Let me give you one more example before I wrap up. The idea of a molecular motor, right? We know that proteins in the cell, some proteins in the cell like kinesin and myosin, are sort of responsible for moving cargo from one place to another, and sometimes because of this mechanicist tradition, they are viewed or described as ingenious nanomachines which convert chemical energy into work. It is argued often that they undergo these power strokes similar to the ones that you find in an internal combustion engine of your car, which result in periodic conformational rearrangements driven by cycles of ATP. And so often this is represented as sort of cycles of walking. So these robotic bipeds that walk across the cytoplasm in the microtubule, and they take cargo. So this is just this is often represented. I've got a little video to show you, which you've probably already seen. So, it gives you this idea that everything is organized, everything is stable, and, the point is that no one has actually seen this happening. This is a projection or interpretation of empirical data, and it's a problematic one because it fails to take into consideration the sort of stochastic environment that these proteins actually exist in, okay? This is just some examples from the literature. These are, from review articles showing how the scientists are sort of aware of the problems of moving from one scale to another, saying that, for molecules, moving deterministically is like trying to walk in a hurricane, It's impossible to go in a straight line. The idea that you've got Brownian storms that are raging continuously. So you got this idea again that the notion of machine sort of almost licenses this blindness towards the importance of scale, which is, I think, quite problematic. There's an alternative model to explain the same phenomena called the Brownian ratchet principle, which does not require to assume any sort of structural rigidity or specificity, and gives you the same product. It's actually a really good example of order from disorder, to take another phrase that came up, but I'm not going to discuss it because I don't have time. Okay, so I just finally want to ask this question. I mean, why is it that, that molecular biology embraced this mechanical view and dismissed stochasticity? I mean, this is something that is historically very important for us to understand, because this view of the cell was never inevitable. Things could have been different. If you look, for example, at the writings of David Bohm, prominent quantum physicist, did remarks in the late 60s that it really seems odd that just when physics moved away from mechanism, as it did as a consequence of the quantum revolution, biology has been moving closer to it, right? Saying that it's going to lead to this sort of schizophrenic situation where scientists will be regarding living beings as mechanical while supposing that inanimate matter is too complex and too subtle to fit into the categories of mechanism. So why is it that physics gave up mechanism and biology came closer to it? Another quote from Carl Rose saying basically the same thing. Biology of the 20th century was in this strange position of having to contort itself to conform to a worldview that physics had already abandoned, right? And so I think this is important to realize, because it makes everything sort of, nothing is inevitable. We can sort of, take a step back and realize that, that where we think about this cell in this mechanical way that leaves no room for randomness is not inevitable. And I'll finish just by suggesting that part of the reason why we ended up in this situation has to do with the legacy of this book, which has come up several times already, Erwin Schrödinger's What is Life, where, this book is really about order, about the nature of order, and he suggests that unlike the order that you find in physics, this order from disorder, right, this sort of statistical mechanical idea of order, where you have the law of large numbers applies, and you've got regularity coming in that way. The order of life is based on this idea that it's all based on a single molecule, the aperiodic crystal, the genetic material that is fixed and has a solid state, and because it's fixed and it has a solid state, it is impervious to stochasticity. Stochasticity does not have to be taken into consideration because the structure of the genetic material is sufficiently solid and rigid that we don't need to worry about it. And what I think happened is that idea was passed on to how we think about proteins. It's not just DNA that are understood to be as crystals, but also proteins, So that means also we don't need to worry about stochasticity because everything is internally determined in a way. And so I think this is my hypothesis, that the influence of this idea granted molecular biologists a license to not have to worry about randomness, not have to worry about stochasticity, allowing them to focus on the structure of the macromolecule, drawing attention to the crystal-like rigidity, emphasizing the functional specificity, and ignoring the destabilizing influences. And if you're interested in this, stay tuned, because I'm currently writing a book on this, which will come out next year, called What is Life Revisited, which I'm basically developing this argument at length. So my conclusions. Molecular biology remains philosophically committed to many of the tenets of this mechanicist worldview of the scientific revolution. That's really the only way we should think about how reductionism and determinism fits into that picture. It's grounded in that view. This conception is problematic for many reasons. The one that I've discussed today has to do with the fact that it inappropriately projects features of the macroscopic world of our machines into the strange microscopic world that our molecules exist in. Importantly, the impact of stochasticity, this is not new. We've known this for a century, okay? This is not a new development. But it's been not known for a long time. The Darcy-Thompson quote is from 1917. What I want to suggest is it is the influence of people like Schrödinger and his What is Life that led us to sort of momentarily forget what we used to know. And that's it. Thank you very much. These are just further readings if you're interested in following up these arguments in more detail. Thanks.
Thank you very much, Dan, for this very thorough analysis of the issue of mechanicism in biology. And I think you made a very convincing point on the difficulty of the difficulty of applying mechanism at the molecular level. So we have time for a few questions.
You just opposed the biological scale and the mechanical scale, but in what extent, these two scales can interact together? For example, in the biomechanical concept. Could you explain this, please?
That's a great question. In fact, there's a debate that happens in the 1930s among physicists about exactly this question. Should we consider the indeterminism of quantum mechanics to play a role in how we think about the cell, for example, right? So is there sort of an, and there are people like Pascual Jordan is one of the of Bohr's students who suggests that, you know, that actually cells and organisms are actually indeterminate because they serve as amplifiers of quantum effects, right? So the idea there is that you've got a connection between the quantum scale, really, and then the biological scale, okay? And then he actually coins this term of this idea of quantum biology that then is sort of forgotten and only now is beginning to, you know, experience a renaissance. But someone like Schrödinger actually had a very different view. So for him, quantum mechanics was important because not because it showed indeterminacy, but because it showed the fixity and stability of the covalent bond that connects the atoms in the genetic material, right? So. So essentially, he's using quantum mechanics in the opposite way. So to show that the macroscopic scale has to be determinate and fixed, right? And that the effects of the macroscopic scale, Brownian motion, can be eliminated because these molecules are so rigid and so fixed that they can withstand that disorder, right? So, but I think that question today, I mean, so that's a historical answer to your question. A philosophical or scientific answer is that that's something we're still working out now. To what, you know, in which ways, right? Phenomena or processes that take place at the molecular level, which may be indeterminate, end up influencing how we think about, you know, the phenotype. I mean, a good example is the emergence of variation. You know, the role of noise in developmental biology would be an example of how something that happens, you know, gene expression is stochastic, and yet it seems to have an effect at the macroscopic level. So thank you for that question.
I have just a curiosity, because you gave a very convincing argument about the fact that a cell like a machine doesn't work, and you gave some reason for that, and also the fact that the molecular machine overemphasizes structural rigidity, functional specificity. So my question is, maybe in your next book, do you have an ontological proposal about the way in which we have to rethink about cell ontology? It's kind of, okay, we only need to structural flexibility, functional plurality, or do you think to give a more strong overview about a new ontological self-view. Thank you so much.
All right, thanks for that question. Actually, I'm very glad to tell you that I have that, and I have that in the book in my previous book called Everything Flows. And it's a book, it's called Everything Flows Towards a Processual Philosophy of Biology, published with Oxford University Press. It's open access, so you can download it for free if you just Google it. Everything Flows and my surname. And actually, what I do in this book, it's not just my book, it's a collection of essays, and I contribute several papers to that collection, is to suggest that process metaphysics, process ontology is better suited to biology. That instead of thinking about biological entities as substances, which conforms to the way we think about them as machines, maybe we should think about them as stabilized flows of energy and matter, right? As dynamically stable processes. And if it requires you to sort of undergo a Gestaltschrift and think about the world, the biological world in a different way. But once you do, everything begins to make sense, right? So 18 scholars contributed to this book, and I encourage you to read it. Basically, we sort of propose that process thinking can give us an alternative ontology for life that can basically save the phenomena. Because at the end, what we want to do is do justice to the science, right? And so we think that an ontology of process is far better suited than an ontology of machines to make sense of what's going on today in molecular biology.
Le hasard va-t-il tuer l'animal-machine? J'ai l'impression qu'on pourrait dire que la présentation de Dan Nicholson vraiment le souhaite, puisque, comme il l'a dit, depuis le XVIIe siècle, finalement, on pourrait presque dire qu'on est dans un paradigme mécaniste dans la biologie, puisqu'on considère qu'à peu près tous les niveaux biologiques peuvent être considérés comme des machines, des organismes jusqu'à aujourd'hui, les cellules que la majorité des gens peuvent considérer finalement comme des machines, la cellule pouvant être vue comme finalement une espèce de collection, d'ensemble, d'associations de protéines considérées elles-mêmes comme des machines. Le problème, et c'est ce que Dan Nicholson a montré, c'est qu'une telle considération de la cellule comme une machine met trop en avant la rigidité structurelle dans cette idée. Elle oublie le fait que la configuration structurelle dépend du contexte. Et de même, cette conception met trop en avant la spécificité fonctionnelle, alors même que les associations sont normalement transitoires et contingentes. Donc finalement, il semble qu'on devrait pouvoir espérer que la question du hasard va remettre en cause ce paradigme mécaniste, d'autant plus que, et d'ailleurs il y a fait rapidement référence au début, Descant finalement, avec l'opposition à la notion du vivant et de la montre, on voyait bien par contraste qu'un vivant s'agrandit en se différenciant. Ça ne grandit pas comme un assemblage de parties parthèses, extraparthèses. Au contraire, ça part de quelque chose qui se complexifie au fur et à mesure. Or, cette complexification progressive, elle semble pouvoir être prise en compte dans une ontologie processuelle, notamment, mais est beaucoup plus difficile à appréhender dans un paradigme mécaniste. Donc, finalement, espérons que cette ontologie processuelle et par-delà aussi la question du hasard permettra de remettre en cause le paradigme mécaniste. Merci beaucoup et merci à Dan Nicholson.